For my project, I explored the impact of elapsed burn time on restored prairie plant biodiversity as well as soil nutrient content. So, for a little bit of background, I decided to study prairies because they are one of the most endangered ecosystems in North America, and we only have about 1% of our natural prairie existing today. And this is a really big problem because prairies are a super important ecosystem. They house dozens of rare native plant species and animal species, and so, given their importance and how endangered they are, a lot of emphasis has been placed on finding the best methods of restoration so we can try and restore more of our natural prairie. I focused on tallgrass prairies in Minnesota in an attempt to explore the environmental consequences of one widely used method of habitat and prairie restoration, which is prescribed burning. So essentially, as you can see in the picture, prescribed burning is just when a section of prairie or other habitat is set on fire, and then this is really effective for restoring prairies because a major threat to prairies is invasive species, and so this clears the invasive species, and the native plants grow right back because they have really long roots. So I wanted to explore how the amount of elapsed burn time impacts plant biodiversity and soil nutrient content in restored prairies because both plant biodiversity and the nutrients in soil are important indicators of how healthy a prairie is. So I thought these would be two variables that would be a good indicator of how prescribed burning is affecting the health of restored prairies. I hypothesized that the plots of prairie with the shortest elapsed burn times would have the greatest amount of plant biodiversity, as well as the highest concentrations of nitrogen and phosphorus within the soil. So, in order to conduct my study, I looked at sections of restored prairie located at the Cowling Arboretum, which is the Arboretum of Carleton College in Northfield, Minnesota. And I picked three sections of prairie because, as you can see on the map on the right, there are different management units, and each was burned at a different time. And so the three sections I picked had varying amounts of time elapsed since they were most recently burned. But other than that, they were all pretty similar in terms of their burn history. And so I tried to keep many things about them constant. But the three I chose are referred to as B2, D2, and D3, and they were burned half a year, 1.5 years, and 6.5 years ago, respectively. And you can see the three sections that I sampled from are starred on the map. And so, to sample plant biodiversity, which was the first variable I was measuring, I constructed a 1 by 1 meter frame, as you can see on the right, and that was placed on the ground in three random locations within each section of prairie. So 9 quadrants were sampled from total. And essentially, I just counted the total number of species as well as the number of individuals within each species because I would use both parts of that data in order to perform my analysis. And then the second part of my study was looking at soil nutrient content. So I collected soil samples from each section of prairie, and I collected 3 soil samples from within every quadrant that I measured biodiversity in. So there were 9 samples per plot with 27 total. And so after I collected the samples, they were brought to the lab where I used the Lamont Soil Nutrient Test Kit to analyze both the nitrogen nitrate concentration as well as the phosphorus concentration. And I looked at these because they are important indicators of how healthy soil is. And then in order to analyze my data, for the first part, which was the plant biodiversity, I standardized my data that I collected in the field on the number of species and number of individuals using Simpson's diversity index because it's really nice in that it gives you one value that's easy to compare across plots. And so that's the lab entry I included on the right, showing my process of using that Simpson's diversity index to come up with my ultimate diversity scores for each plot of prairie. And then for the soil data, it was pretty basic. I just averaged the concentrations I found when doing the soil testing and constructed bar graphs with error bars and tested significance with a one-way ANOVA. So here you can see the first part of my results with the impact of elapsed burn time on plant biodiversity. So as you can see from the bar graph, I found that biodiversity was greatest in the two sections of prairie burned most recently. So these had the least amount of elapsed burn time, and that's sections B2 and D2. And you can see that they're significantly higher than the plot of prairie that was burned least recently. Unfortunately, I could not obtain a p-value for this data just based on the way the symptoms diversity index works. So that would be an area for future studies to test if this relationship holds true with multiple trials. And then moving on, here is the impact of the elapsed burn time on my soil phosphorus content. So similar to the plant biodiversity results, you can see that the phosphorus concentration was highest in the plot of prairie burned most recently, so plot B2, with a concentration of 47.22 parts per million, and this was significantly higher than either of the other two plots' concentrations, and this was significant with a p-value of 0.005. And then the final piece of results was on the nitrogen nitrate concentration. And so this was where the data did not support my hypothesis because, unlike the other two relationships I determined, for this one, I actually found that nitrogen and nitrates were highest in plot D3, which was burned, had the greatest amount of elapsed burn time. And this had 15 parts per million, which was, as you can see visually, much higher than either of the other two plots. And this finding was significant with a p-value of 0.001. So what my results indicate is that the plots of prairie that were burned more recently are generally healthier because they did have greater plant biodiversity as well as higher concentrations of phosphorus in the soil. However, there was that one piece of data that did not fit with what I was expecting with the nitrogen nitrate concentration. And so that's something that I would want to examine more in the future to just do more tests and see if that relationship was an anomaly or something that we could expect with multiple trials. And all the findings were significant that I could determine p-values for, so the results were significant. So what these results mean in context is that restored prairies burned more recently will generally be healthier based on the fact that the plant biodiversity and phosphorus concentrations were higher. And this is super important and useful to habitat specialists and people who are focused on restoring tallgrass prairies because they're always looking for more research and more information on the best practices for efficiently restoring habitats that are endangered like the prairie. So some future work could definitely be useful to this experiment. I would definitely like to replicate the same experiment with more trials just to determine if the relationship is still going to hold true with a greater sample size. And additionally, I could perform a study to determine how burn frequency impacts other factors, such as the concentration of other macronutrients in the soil, or I could also explore how burning impacts plant biodiversity, but pay more attention to the native plants that I see versus invasive species, because that's an important distinction that biodiversity is not always a good thing if that includes harmful species to prairies. So those are all just some ideas that I could definitely explore further. And there are my references. Thank you.